In this slide set, we're going to talk about techniques that purify, detect, and characterize protein. This is for either diagnostic reasons, directly for diagnosing diseases or such, or for understanding biochemical pathways, or for further use of proteins in drugs. There are many applications for these techniques, and they need to be well understood. As you will see in the near future, the other common macromolecule that is in your body are nucleic acids, mainly DNA and RNA. And when we are going to discuss their recognition and understanding their structure, it's not very complicated when compared to proteins. The reason for that is that proteins vary in size, they vary in charge, and they vary in water solubility. Another complication is that there are 10,000 different proteins for each cell. I'm not saying of how many molecules there are in the cell. There are 10,000 different protein types inside the cell. Their quantity differs from one protein to the other. In addition to that protein separation is usually based on size or a specificity to a ligand, as opposed to straightforward techniques for DNA recognition, such as PCR, for example. Centrifugation is the simplest technique for protein separation. Its principles depend on having two different particles in suspension. These different particles should have either different masses or densities. Those will settle to the bottom of a tube at different rates. All proteins vary in mass, but because, as we learned previously, they are all made up of the same amino acid, their density, which is their mass over volume, is pretty much the same. So in the case of centrifugation, we are going to differentiate between proteins based on their mass. These different proteins with different masses are going to sediment, so they're going to move to the bottom of a tube where they are in suspension at different rates, even if we do not apply any external forces to these masses. However, in order to make the process a lot faster, to expedite that sedimentation process, we subject the tube with these proteins in suspension to centrifugal forces by spinning the tube where the proteins are present. Usually, centrifugation works for particles that are larger than 5 kilodaltons. A dalton is a measure of mass, and it's about 10 to the minus 24 grams, so it's very, very small quantity. The purpose of centrifugation is either as a step to prepare further investigations into the identity or the structure of a protein. So we separate different proteins and we take the ones we are interested in, and then we further analyze them, or as a direct analytical technique to measure physical properties of a protein, such as its molecular weight, its density, its shape, and the equilibrium binding constant. The measure of centrifugation is by measuring a sedimentation constant of a protein. So that's the property of the protein which centrifugation differentiates proteins based on. So you measure those sedimentation speeds by measuring sedimentation constant, and its unit is the unit Svedberg. The first type of centrifugation is differential centrifugation, and in general it's used to separate soluble from insoluble proteins. So a sample is poured in a tube. The sample contains larger and smaller particles. Usually the larger particles are insoluble, but in any case, the tube is subjected to centrifugal force. Centrifugal force is directly proportional to the mass of the particles inside the tube. So larger particles, once centrifugation is stopped, the larger particles condense at the bottom of the tube, forming an aggregate called a pellet, and the remaining smaller particle proteins that are usually still in solution remain in what we call the supernatant. The second type of centrifugation is rate zonal centrifugation. It's very similar to differential centrifugation. However, the basic difference is that there is a density gradient that's added to the tube. That density gradient is added through pouring a solution of increasing density inside the tube, then pouring the mixture in which the protein we want to separate on top of that solution. Usually the density gradient solution is made of concentrated sucrose, and it's very important to take into account the time we subject the sample to centrifugal force, because if we spend a lot of time subjecting the sample to centrifugal force, no separation is going to take place and all the particles are going to sediment at the bottom of the tube. Please note that rate zonal centrifugation is not effective in determining the exact molecular weight because sedimentation not only depends on the mass, as we discussed earlier, it also depends on differences in protein sizes, which is mostly shape dependent. So as I just mentioned, there's a sucrose gradient that's already inside the tube. That gradient has a very high density at the bottom of the tube, and as we move to the top, that density decreases. The protein solution is poured on top of the density gradient. Then the centrifugal force is applied onto the tube, and the particles settle at different zones according to their mass. Particles with the highest mass move towards the bottom, while the lightest particles move towards the top. So when centrifugation is stopped, we end up having more than two fractions as opposed to differential centrifugation. Here we'll have multiple fractions. The bottom fraction will have the heaviest particle mass, and as we move to the top of the tube, the mass of the fraction would decrease. Equilibrium density centrifugation is similar to rate zonal centrifugation. However, it differs in the fact that we are not talking about separation of protein, but about separating other larger molecules that have different densities, because as we mentioned earlier, proteins have similar densities, so centrifugation is going to separate them based on mass. But if we talk about other molecules that differ from one another in density, we also present a gradient, density gradient. And since we have different particles that not only contain amino acids, but also they contain nucleic acids, proteins, lipids, sugars, and sometimes organelles, that means that these entities have different densities, and once we present them in a tube and apply centrifugal force to them, and that tube has a density gradient, that they're going to separate according to density. Organelles. Here we also have a density gradient also made up of sucrose. However, the gradient is larger than that of the previous centrifugation process that we described. Again, increasing density gradient. So when we move to the bottom, the density is higher, to the top it is lower, and then we subject the tube to centrifugal force and organelles and larger particles. So all of these are much larger than a protein, are going to stop at different density values depending on their density. And the reason why their density differed, as I just mentioned, is that they are not proteins, they are larger macromolecules that are not only made up of amino acids, they are also made up of lipids and sugars. So their densities can be separated clearly using a sucrose gradient. So in this case, the separation is based on density. In the case of proteins, they are separated based on mass. Now that we're done with chromatography, a second separation technique that we are going to discuss now and then discuss again when we talk about the separation of nucleic acids in a couple of months is electrophoresis. Electrophoresis, we can understand the meaning of the word if we separate it into two parts. Electro involves applying an electric field or electric current. Phoresis means movement, so we have movement of particles when they are subjected to an electric field. So if we have a mixture of molecules and we apply an electric field to them, the E field is going to move the particles at a speed that depends on the particles' charge to mass ratio. So the smaller the mass, the larger the effect of the electric field, and the greater the charge, the greater the effect of the electric field. Electrophoresis is applied on proteins using a technique called SDS, polyacrylamide gel electrophoresis, or in short PAGE. SDS stands for sodium dodecyl sulfate. Of course, it's called polyacrylamide gel electrophoresis because the electric field is subjected in the protein mixture after it is placed in a polyacrylamide gel, and as I mentioned, the electric current is applied to the gel. So once the current is applied to the gel, the proteins are going to move, as we just mentioned, according to their charge to mass ratio. So smaller proteins, because their mass is a lot smaller, are going to travel faster through the gel. And the purpose of the gel is that it acts as a sieve. So a sieve is what is used to the kitchen to purify things. So if you add water and rice in the sieve, the rice stays in the sieve and the water moves out. That's what a sieve is. There's also a shape effect because the resistance of the gel to the protein is going to depend on the protein's shape. Of course, to control the protein movement in the gel, we can change the pore size, which we can control and change by adjusting the concentration of the polyacrylamide and the cross-linking reagent for the gelation process. Polyacrylamide gel, if you remember when we talked about hydrogels in biomaterials, is a hydrogel, and for hydrogels to become a hydrogel, they need the formation of cross-links between the different polymer chains. The more cross-links there are, the smaller the pores. So we can control the pore size by controlling the concentration of the polyacrylamide. The higher the concentration, the smaller the pores. And the cross-linking, the more cross-linking there is, also the smaller the pores. If you know that your mixture has large proteins, there is no need for having very small pores. We can have larger proteins. However, if you have smaller proteins or proteins that are close in size to one another, then we need to have smaller pore size gels. The rate at which proteins move through an electrophoresis gel is dependent on two factors. The gel's pore size, which we can control by controlling the cross-linking in the gel. So the more cross-linking we have, the smaller the pore size, and the smaller the pore size, the more resistant it is for the movement of the protein, so the slower the movement of the protein in the gel is. And the strength of the electric field is the other factor, and the stronger the electric field is, of course, the greater the force by which the protein is pushed through the gel, so that would make its movement through the gel faster. Proteins can be exposed to ionic detergent SDS before and during PAGE. Of course, that is to both denature the protein and to provide it with a negative charge, so it would only move in the gel based on its molecular weight. The mechanism by which SDS denature the protein is by binding to its hydrophobic side chains. That binding would disrupt the hydrophobic bonds that put together the protein's 3D structure, destabilizing the hydrophobic interactions of the protein, and this would cause it to use its 3D structure and to destabilize its conformation, bringing it back to its primary polypeptide chain structure. So in addition to using SDS to denature the protein, heating and a reducing agent are also added to the protein in order to further denature it and make sure that it has no secondary and tertiary structure. So as I mentioned earlier, the treatment with SDS eliminates the effect in differences in charge, in the native shape, in the native structure, making the change length the determinant of the migration rate. So it only depends on how long the polypeptide chain which makes up the protein is. The molecular weight of the protein in SDS page can be estimated by comparing it to the migration distance with a known protein of a known molecular weight, which is called a protein ladder. And afterwards, proteins that run in a gel that we can estimate their mass with can be extracted from the gel and used for further analysis. We can see here we have a protein sample. That protein sample is subjected to heat and also to SDS in order to denature it. So we have an SDS coated protein. All the proteins then are denatured and have a negative charge. The sample is loaded onto what we call different wells. So you can add here 1, 2, 3, 4, 5, 6, up to 7 sample. This is the shape of the polyacrylamide gel. And since all the proteins are negative, we apply an electric field so that the protein would move from a negative charge to a positive charge. So all the negative proteins are going to be repelled from the negative charge and are going to be attracted to the positive charge. What happens is that the proteins are going to migrate in this direction towards the positive side of the gel, and they are going to separate. Proteins with a larger mass are going to have greater resistance from the pores in the gel, so they're going to move the slowest. Proteins with the smallest size are going to move more easily and cross the pores within the gel, so they're going to travel the furthest. So here, after we run the whole gel, so we subject the gel to an electric field, sometimes between 45 minutes to hours, 2 or 3 hours, depending on the protein and the resolution we want to get, how much separation we need. So at the end, we're going to get different sized proteins inside the gel. So this is one sample, and this sample has 1, 2, 3, 4, 5 different sizes of proteins. And as I mentioned earlier, the further the protein travel, the smaller its size. This is a cell lysate, and the cell lysate, that's when you bring a bunch of cells and you break them down. So you bring a bunch of cells, you break them down, and then you add the sample to a well, so you get multiple proteins. And you can see that by a lot of bands here that you can it's very difficult to differentiate between them. A purified protein, if you run it on a gel, it's only one single protein. You might have purified it using one of the centrifugation techniques we mentioned earlier, and you add it to a well, then you're going to get one single band. This is a protein ladder, so we can estimate the size of the protein depending on where in the ladder it corresponds. So this purified protein is 66 kilodaltons big. This protein is about 40 kilodaltons big. So this is the ladder that we mentioned before, which is a bunch of proteins with known molecular weights. A more sophisticated type of gel electrophoresis is two-dimensional gel electrophoresis. So as we mentioned, page separates protein based on mass. But we know from our previous studies that there could be many proteins that have a similar mass. So page is unable to separate these proteins. They're going to be in one band and you won't be able to recognize them using page alone. So to further complicate gel electrophoresis, 2D gel electrophoresis was developed, and as I just mentioned, it's used to resolve proteins with similar masses. So in addition to mass, we're going to differentiate their proteins based on their charge. So it uses the difference in electric charge, which is determined by the pH, and the relative number of proteins' negative and positive charge groups. I'll explain in a bit. So in 2D gel electrophoresis, we firstly separate the protein based on its charge. Afterwards, we separate it based on its mass, which is exactly identical to the page part that we discussed earlier. So in two-dimensional gel electrophoresis, proteins are denatured, but they are denatured in urea because, as I mentioned earlier, SDS adds a negative charge. But since we want to separate the proteins based on their charge, we cannot eliminate that charge. So we need another denaturant to eliminate the effect of shape. In this case, urea is used. Afterwards, a pH gradient is formed by a material called amphilites, which is a mixture of polyanionic and polycationic molecules. So some of those molecules are anionic and they have a positive charge, and others are cationic, so they have a negative charge. So the charged protein is going to move along that gradient until it reaches what we call an isoelectric point. The isoelectric point, or PI in the slide, is the pH at which the net charge of the protein is 0. So the first dimension of separation in 2D gel electrophoresis is called isoelectric focusing. The second part is electrophoresis. So an isoelectric focusing strip, after the proteins are separated based on their charge, the strip is transferred to a gel with SDS for the second dimension, which is electrophoresis. Here we can better explain two-dimensional gel electrophoresis. So as I just mentioned earlier, the first dimension is isoelectric focusing. Isoelectric focusing takes place when we present amphilites to make up a pH gradient to which the protein mixture is added. The pH gradient has a very low pH where hydrogen atoms concentration is high, giving it a positive charge, while a low pH has a very low hydrogen concentration, giving it a negative charge. As you know, all proteins have a deprotonated and a protonated state. So let's say if they have an O group at its end, that is going to give it a negative charge. So this protein, if it's added here, it has a negative charge. It's going to move towards the positive end. As it moves towards the positive end, any negative charge it's going to move at an increasing concentration of hydrogen atoms. So the deprotonated parts are going to get protonated, and the protein's negative charge is going to become smaller and smaller until it reaches a point where it has no charge, it's neutral. On the other hand, if we add a protonated protein that has all its positive hydrogen atoms, because it's positive, it's going to move towards the negative end. As it moves up the pH gradient, so down the hydrogen concentration, it's going to start losing its protons. So it's going to get less positive until it reaches a point where it has a neutral charge, and it won't move towards neither the positive nor the negative side, and it's going to stop at this spot where it has a neutral charge, which is, as I mentioned, called the isoelectric point. This strip in isoelectric focusing, after we separate the proteins based on charge, is going to get transferred to a page tub. The page tub is going to have SDS in it. So all the proteins are going to gain a negative charge. We've separated them already based on their charge. Now we know where the spot for each one is. So we don't need to know their charge anymore, but we still need to separate them based on mass. So we present them in a tub that has SDS in them, and we again apply an electric field. The proteins are going to move from the negative end to the positive end, and the further the protein moves, just exactly as it is in page, the lower its mass is. So even though this gel looks similar to what we've seen before in page, not only do we separate the protein in the y direction based on its mass, we had already separated it before in the first dimension based on its charge. For example, if we look at these two proteins have the same charge, however, they have different masses. On the other hand, these two proteins have the same mass, but they have different charges. If this were page without the first dimension, we're going to have separation for these two proteins. However, these two, the second ones, the ones that are only separated based on charge, are going to be in the same band. This is an example of an SDS page. You can see it here on the right side of the screen. This is a typical SDS page, and as I mentioned, this dimension is going to separate based on charge, where this dimension is going to separate based on mass. The third separation technique we're going to talk about in this slide set is liquid chromatography. We've briefly mentioned liquid chromatography in the biomaterials class, in what was similar in gel permeation chromatography. Their principle is exactly the same, however we can explain it again here. So the principle of liquid chromatography is that molecules in a solution associate or separate differently with particles in a solid phase, which is dependent on the physical and chemical properties of the molecule and the surfaces. So if a solution is flown across a surface, the molecules in the solution are going to interact with the constituents of the surface, and that would let them spend more or less time in that interaction, either slowing down or fastening the flow. The nature of the beads that make up the solid surface with which the components of the solution are made of determine whether the separation of the protein is dependent on their mass, their charge, or their binding affinity. So here in this slide set, we're going to talk about three different types of chromatography. The first type is the simple gel filtration chromatography. Here the separation is based on size, where the solid surface we just talked about has a column that's composed of porous beads. So small proteins in a solution are going to take longer because they have more options and a longer path to travel through than larger proteins. The total volume of the liquid required to elute a protein from a gel filtration column is also dependent on its mass. The smaller the mass, the more liquid we need, so the more elution volume it's going to need and the more time it's going to need. What happens in gel filtration chromatography is that the protein solution is placed in a column where there are beads that are going to present different sized pores in the column. Once the solution is poured, the different sized proteins are going to start finding their way across down the column due to gravitational forces. So what happens is that smaller molecules are going to have longer paths to travel because they can go through smaller pores, and that's going to make them go along a longer route, and that is going to cause them to have a longer distance to travel. While larger molecules don't have many options of where and how they're going to go, so their paths are a lot shorter. So for larger molecules, they're going to go directly down the one path. So their path is much shorter. Larger molecules' path is much shorter. That is going to take less time for them to travel, meaning that the first fraction coming out of the column, once the fractions are collected, are going to be larger proteins, and the smaller the protein is, the more time it's going to take to travel down the column, so the fraction is going to be the last one to be collected. Larger proteins collected first, smallest proteins collected last. In ion exchange chromatography, the proteins are not separated based on size, but they are separated based on charge differences. Here in this case, the beads are modified with either carboxyl or amino group. Carboxyl group present negative charges on the beads, while amino groups present positive charges on the bead. In a positive bead column, so the one that is treated with amino groups, only proteins with a negative charge are going to interact with the beads, while positively charged proteins are going to be repelled from the beads. After the positively charged proteins go through the column very quickly, negatively charged proteins are eluted by passing a solution that has an increasing concentration of salt. That would cause the salt molecules to interact with the beads and replace the negatively charged proteins, which would cause them to elute down the column. So in this case, the first proteins to be eluted are the ones that are the weakest. So in the case of a positive column, the most positive proteins are the weakest bond, while the strongest bond is the most negative. Again, we have this column. The column has proteins that are negatively and positively charged added to it. The beads themselves are positively charged. So the positively charged proteins are going to go through quickly because they are going to be repelled by the positively charged beads. So the first fraction is going to be the most positive, as I mentioned earlier, and the negatively charged proteins are going to bind to the column. So in order to get these beads out, the negative ones, we add a strong salt solution to the column. This salt solution replaces the proteins and causes the negatively charged proteins to elute down and be collected at the end of the column. The last type of chromatography we're going to discuss is affinity chromatography, which is based on the ability of a protein to specifically bind to another molecule or another protein. We defined molecules that bind to the protein by the term ligands. These ligands are covalently bound or attached to the beads in the column instead of adding a positive or negative group, as we mentioned in ion exchange chromatography. In this case, only one protein binds to the column, which is the protein specific to the antibody or ligand that's bound to the beads. After the protein is bound to the beads through the antibody, what happens is that we elute that specific protein by either adding an excess of the soluble form of the ligand, which is going to bind to the protein instead of the ligand that's bound to the beads, or by changing the salt concentration or pH, both of which are going to change the conformation of the protein or its ligand, or both, in which case the bond between the two would weaken, and that would cause the protein to elute out. Again, so what we have is a mixture of proteins. So the red ones are proteins that are specific to the ligand that's bound, and in this case it's an antibody that's bound onto the bead. So this is the protein that's specific to the antibody. And the blue ones are other proteins that won't bind to that antibody. Once the mixture is added, all other proteins are going to elute out, except for the proteins that are specific to the antibody. They're going to bind to the beads through binding to the antibody. We elute the protein that is bound to the antibody by changing the pH and adding a buffer with a much lower pH. The regular pH was 7. Adding a buffer with a pH of 3 would cause a protein change in conformation and an antibody change in conformation. This would weaken the bond between the two and cause the protein to elute out of the column.